Okay, so let's talk a little bit about platelets or thrombocytes. So platelets, very similar in the way red blood cells are stimulated by erythropoietin. Platelets are stimulated by thrombopoietin. So that thrombo prefix, so that thrombo base really means platelet, right? So it's a platelet cell. They don't live very long, so they only live about 5 to 9 days. Similar to red blood cells, macrophages, fixed macrophages in the spleen and the liver, are going to remove them and recycle them. And their main role is to help stop blood loss from damaged vessels by forming a plug and then forming a clot. And we'll talk about that process in a few slides. Now, although they are tiny, tiny little fragments of cells, you can see here a megakaryoblast becomes a megakaryocyte, and then that breaks up into these tiny little platelets. These tiny little platelets are anything but hollow or empty. There's all of these granules inside them, as well as organelles, so they're actually really complex on the inside, but otherwise super, super tiny. If you remember, a red blood cell is 8 microns in size, so these are much smaller. Again, you don't have to memorize these numbers or anything like that, but there are more platelets than there are white blood cells, but not as many as red blood cells. All right, so let's talk about their main role. So how do you form a blood clot? And by the way, this dove has not been injured. It is called a bleeding heart dove. It's found on a small island in the Philippines. Naturally looks like that. It is a near threatened species. But anyway, I thought it looked kind of cool and went with this chapter, so there you go. Okay, so let's talk about blood clotting. There are 3 main methods for hemostasis, or the stoppage of bleeding, and that is vascular spasm, platelet plug formation, and then blood clotting. So let's just take a quick look at this image here on the right. So here we can see the site of injury. The blood vessel is going to contract, and simply by contracting or vascular spasm, the flow of blood is reduced, and therefore the loss of blood is reduced. This also allows for the aggregation of platelets, so they can more easily form a little plug. And then a clot, a fibrin clot, is sort of a protein mesh on top of the plug that makes it really strong and tight. Something I want to bring to your attention is this term coagulation. It is different from agglutination that we saw with red blood cells. So coagulation is the formation of fibrin threads. Agglutination is an antibody-antigen complex. So even though you'll have the clumping of cells in both cases, agglutination that we saw in red blood cells is due to antibodies attaching to antigens and clumping cells together. Coagulation, on the other hand, results in cells being clumped together and held together by fibrin threads. So they are different. Please make sure you distinguish between those 2 terms. Okay, so first of all, vascular spasm, simply the radius or the diameter of the blood vessel is reduced, again, reducing the flow of blood. It is basically in response to pain receptors, so it's a reflex, and again, can reduce blood loss until a proper blood clot can be formed. This only works for smaller blood vessels. If you happen to have a puncture of something, a large one like an aorta or something like that, vascular spasm is not going to function very well. I mean, you can bleed out and die. A platelet plug, on the other hand, so what happens is, again, these platelets have lots of these chemicals and these granules. There are alpha granules, and then there are dense granules. The alpha granules have clotting factors, so these are different proteins that are going to be important in a very long clotting cascade. There's also something called a platelet-derived growth factor, and this helps repair the walls of a damaged blood vessel, including the endothelial cells, the muscle, and even fibroblasts to repair like the fiber around a blood vessel. Dense granules will contain all of these different things. So ADP, which is basically ATP with one of those phosphates removed. ADP causes the aggregation of platelets, and it induces them to change shape. We'll talk about that in a second. ATP will help recruit macrophages. Calcium is going to be used in every single step of that clotting cascade. There's serotonin. This aids in vasoconstriction and platelet aggregation. Fibrin stabilizing factor will help stabilize fibrin threads. And enzymes and thromboxane A2 will also aid in vasoconstriction as well as platelet aggregation. Again, platelet aggregation is just the grouping or the coming together of a lot of platelets. Excuse me. Okay, so let's talk about platelet plug formation first. So there's 3 steps in the process. There's adhesion, a release reaction, and then aggregation. So when we mentioned just a moment ago that the platelet can change its shape, so under resting conditions it has sort of that little just discish kind of shape to it. And then remember we said change its shape, well, kind of like sploots out, for lack of a better term. And by kind of extending these appendages, it can grab onto other platelets, as you can see in 1D, and aggregate, right? You can imagine if you just stack a whole bunch of these, I don't know, pancake-shaped things, they're not going to really stick well together, but when they change shape to have these appendages that can hold on to one another, kind of more like an octopus kind of thing, they can really clump and form that plug. Okay, so in that first step, that platelet adhesion, there's damage in the walls of the blood vessel and the endothelium that exposes collagen, which causes platelets to stick. The platelets are activated by that sticking, so they extend, they change shape, they start extending projections, making contact with one another, releasing the chemicals we mentioned earlier to help start the repair of the blood vessel wall, as well as help bring in other platelets to help create the plug. And so they stick together. As this continues, they will stick together and then form a nice plug, and that prevents any more loss. So soon after that, a clot will form. So a clot is simply a platelet plug that has been reinforced by fibrin threads, and that's what you see in this image. These are these red blood cells that are now being trapped by these sort of greenish yellow fibrin threads. Now there's a very long cascade of reactions that has to occur, multi-step reaction that has to occur in order for a fibrin thread to form. We are going to simplify that greatly, but basically just be aware, thing A has to happen in the presence of calcium for thing to form thing B, which forms thing C, D, E, F, so on and so forth, until finally we form fibrin threads. So the things we absolutely have to form a blood clot, so these, or another way to say this, the things we absolutely have to form these fibrin threads, we need calcium, enzymes made by our liver cells, substances released by platelets or the tissue, right? We don't want clots forming just random, so we either need the platelets to recognize that there's damage somewhere, or the tissue needs to start the reaction. As well as a vitamin. So this is one of the few times we actually talk about vitamins, and that's vitamin K. It's a fat-soluble vitamin. 4 of the clotting factors, so 4 of the proteins that are part of this cascade need vitamin K in order to be formed. If they do not have this vitamin, they cannot form. That means that cascade cannot continue, which means you cannot have fibrin threads. Okay. That vitamin K is produced in bacteria in the large intestine, so it tends to be readily available unless there's some sort of damage or loss of bacteria or some other factor. Sometimes people may be deficient, in which case it can be supplemented via diet. Okay. There are 3 pathways to form a blood clot. Again, we're going to simplify these first 2, the extrinsic and intrinsic pathways, because they are very, very complex, and you do not need to know this long, complex process. In general, they both of them are going to end up forming something called prothrombinase. So regardless of whether it's the extrinsic pathway path or the intrinsic pathway path, the end result for both is the same, which is the formation of prothrombinase. In the case of the extrinsic pathway, damaged tissue are going to leak a factor called thromboplastin into the bloodstream. We're just going to say a whole lot of complex things happen, which results in factor 10 and factor 5 to form prothrombinase. It's very, very fast. Okay? Very fast. So an extrinsic pathway, this is like you've cut your finger kind of thing. So it quickly happens. The intrinsic pathway, on the other hand, can take several minutes. In this case, the endothelium, so that's the inner wall of the blood vessel, is damaged. Again, those platelets come in contact with the collagen, and they will release factors that initiate a platelet plug formation and fibrin thread formation. It's somewhat less complex, but still a multi-step process. Again, calcium is necessary in each step. You ultimately have factor 10 and factor 5 forming prothrombinase. So again, the main difference, extrinsic, the damage is from the outside. Intrinsic, the damage is on the inside wall of the blood vessel. These two, you can kind of see a little bit in this image. You're either coming from this direction or this direction to form that prothrombinase into something then we call the common pathway, because either, whether it's extrinsic or intrinsic, this now is the same between the two. So prothrombinase, along with calcium, will convert prothrombin into thrombin. Okay, so prothrombinase, that is an enzyme that will convert prothrombin, so just take the ACE off, into thrombin. So the steps here are, again, enzyme, take off the ACE. So prothrombinase works on prothrombin to basically remove that pro part, and then you get thrombin. Okay, and then thrombin, again, in the presence of calcium, will stimulate, will convert fibrinogen into fibrin. So fibrinogen, that O-gen, just take that off. So fibrinogen will generate fibrin threads. And then thrombin will also go on to activate factor 13, which helps strengthen those fibrin threads. So not only do you have the threads, but you make them much stronger. So again, two different pathways, depending on where the damage is or how the blood vessel is damaged. You get prothrombinase, which then converts prothrombin into thrombin. Thrombin takes fibrinogen, turns it into fibrin threads, and then activated factor 13 strengthens those fibrin threads. And now we have a blood clot. I hope that makes sense to you. If not, please let me know, and I'm more than happy to answer any of your questions.